Okay, so let's get to the PowerPoint presentation. Class number 6, transcription factor response. So I'll look at 3 papers. The transcription factor response of the 3 papers are for Levdikov, Seaton, and Pereira. Hang on. The paper by Levdikov is out of structural biology from York in the United Kingdom. The Seaton paper comes from Keen to Seaton from Bernie's lab in Florida, and the review of Mar R comes from a woman by the name of Inoka Pereira from Louisiana State. Okay, so those are the 3 authors that we're looking at. So first paper, Levdikov. Levdikov shows the structure of the COD-Y protein. So what is COD-Y? COD-Y is a dipeptide permease repressor. It controls stationary genes, and there are about 100 of those that it controls. Interestingly, it represses genes that are required for adaptation to nutrient depletion. So when things are starving, it depends on COD-Y. And it could be activated by GTP and isoleucine or valine. So it's being shown to bind to both and be activated by both, but some studies show that GTP is not necessary, like the the Bernie results, and it can be specific for either isoleucine, leucine, or valine. Like some branched amino acids don't work as well as others. So in Bacillus subtilis, it's a dimer, and it's 250 amino acids long. The common term for it is it's a helix turn helix motif, and there's a motif for DNA binding. All right, in the paper, they show a overall structure, and in picture A, I've got the COD-Y protein. Now it's in ribbon structure, and it shows the isoleucine inside in the binding pocket. Letter B is the same protein on its side, and letter C is just in order. So the winged helix turn helix would be right at this end with the wing at the end here.
What's the COD-Y in short mutants? Is there one?
COD-Y.
What's called? Oh.
CODY.
I see. PPGPP regulator.
Yep. Below letters D is the isoleucine, showing that it's hydrogen bonding to all different parts of the protein. And letter F shows valine versus isoleucine. The 2 are modeled. One is, I think, the valine is modeled, but the isoleucine is actually bound in that pocket. So the paper is essentially the protein structure of this protein.
Okay.
That's what the paper is.
Okay.
So we're looking at the structure of the protein. The helix turn helix motif is around the A4, A3, and here's your 2 beta sheets for the wing of the structure. The COD-Y protein also has really good conservation within gram-positives. It's not a gram-negative. It's not really found in gram-negative, it's just in gram-positives, and especially Streptococcus and Bacillus. That's where it's been studied. It forms a dimer. In this picture from the paper, it shows the isoleucine inside of the binding pocket. The next image shows that the COD-Y protein forms what's referred to as a GAF domain. GAF is cyclic GMP phosphodiesterase adenylate cyclase and another protein called FHLA.
Mm-hmm.
So they form a common motif. It's a DNA binding motif, DNA binding domain, and as you can see, it forms kind of like a pocket for the isoleucine. In letter A, it's an alignment of two different proteins, COD-Y, as well as one of these GAF proteins. And in letter B, it's an alignment from the side. Letter C, what they've done is they've put three different molecules in the center. Phosphodiesterase, green, TRA-R, purple, and isoleucine red. Phosphodiesterase is green, cyclic GMP is in there green too. Does that make sense? So they've aligned three different proteins, phosphodiesterase, TRA-R, and COD-Y, as well as their cognate effectors, whether it's cyclic GMP, lactone,
So the
or isoleucine.
phosphodiesterase must really nicely overlap here with the COD-Y,
Right.
because you barely see any green aside from these.
Well, they've done the structure alignment, and by structure alignment, that part sticks in the same place. Yeah.
Yeah.
The effector stays in the same is in the pocket.
Yeah. But I'm just saying everything aligns so nicely that you don't actually get to see the green, right?
Right. In letter D, they're putting in both isoleucine as well as cyclic GMP. I believe that's what they're doing here. That way they show you that that's a binding pocket for that molecule. All right, next picture is a proteolysis of COD-Y, and they did this to try and figure out is it structurally active, and what they're doing is they're adding branched amino acids. So without branched amino acids is on the left, and with branched amino acids is on the right. Top A and B is COD-Y, E and F is COD-Y again. A, B, C, and D is done with trypsin, and E through H is done with chymotrypsin. So trypsin, it cleaves off at the positive residues, whether it be lysine or arginine. And as you can see, you can get around 6 fragments. 1, 2, 3, 4, 5, 6 fragments from that. And if you add branched amino acids, the binding prevents the fragmentation.
I see. Prevent some. Yeah, okay.
Okay. And because BSA is done as their control, negative control, it shouldn't be binding the branched amino acids. There should be no pattern difference with BSA. And with the letters E and F, you can see there's a difference between no amino acids for chymotrypsin, and then when you add the branched amino acids, there's a large difference. So the binding of the branched amino acids prevents the cleavage.
Okay.
Okay.
Cool.
So they're just trying to generate a little bit more structural information that way. Also, what they do is they show you what two other proteins look like when they're bound to DNA, because what they want to do is they want to make the argument that this is a dimer that operates as a transcriptional factor in a similar manner as TRA-R or FAD-R. They're not too sure which way it works. As you can see, TRA-R in letter A, it binds the major groove, and the wing part binds into the minor groove, whereas in FAD-R, it's all located in the major groove. So it's kind of a different binding pattern. So they're not too sure which of the two models it'll prefer. They haven't figured that part out yet. But it's going to be like this. Okay. Next one, the Kinda paper. So Kinda, like I said, she's from Bernie's lab, and what they did was they looked at the transcriptional regulator and its response with the guanosine pentaphosphate and genetic competence. So what do we have? Based on their work, they're calling this the RCR operon. It's because it's rel related, competence related operon, regulated, yeah. So that's why they came up with the RCR name. And there's the two ways of looking at it. There's the origin method, where they have the numbers 835 through 842, I think, whereas the NCBI method labels the same proteins 921 to 928. So this is the more common way. Bernie's group likes to do this, where they show things in the origin format. The paper looked at making deletions in 835 as well as 836 and 837, along with rel-P deletion mutant as well. What is guanosine pentaphosphate? Previous studies have shown that this is an alarm mode that when bacteria are starved, it is activated. So if there's no amino acids, it inhibits the RNA synthesis. Alright, so what do their papers show? They show a couple of tables and a couple of figures. I'll go over the tables first. I think that's how I saved it. So in the first table here, you can see, I think the really best part to look at it is at the end here for transformation efficiency. So 835, they do two mutants. They make a non-polar mutant and a polar mutant. The non-polar mutant will have the kanamycin resistant cassette inserted in a non-polar way, whereas the polar mutant will allow transcription along the regular operon, right? So just go back. Non-polar would probably be in the reverse orientation. So if you have the non-polar mutant, there's no transformation efficiency. And if you have a non-polar mutant for 835 all the way to 37, there's no transformation efficiency. And if you complement it in a non-polar mutant, you still don't get any transformation. And this table goes over kind of the same details. The people also did expression or real-time experiments. Now, these are kind of, I should have printed off the paper. I think this first image shows the expression of 837, and it shows how, no, that's not right, because here's 837. Let me look at the paper for a second. So it's eaten.
It might be expression of competent genes.
This is 36, 836. Oh. I would have thought 836 is deleted in A. So there should be no expression, pretty much, but there is still expression. Letter B is 837.
Oh, I see.
That's correct, right? So this letter A is 837. Sorry, 836. That's shown. 836 expression levels are really low. In A. In B, they're looking at 837. That gene is low for the 836 gene. But 835 is highly upregulated.
Okay. And
And-
in the mutant and the transporters, 835 is highly.
Is upregulated?
Is that what they're saying?
Up there, 835. If you delete 835 in B, then 837 is upregulated. Okay?
Got it. Yeah.
Good. Okay, so does that make sense to you? That's
Yeah,
kinda-
because it's a repressor.
right. All right. Then they looked at the wild type versus the mutant growth. So letter A shows the growth of the nonpolar strains versus wild type. So in diamond, here's the wild type. And as you can see, there's not a lot of difference between the mutants and wild type.
Mm-hmm.
I think I would prefer to have seen this with log scale, because in log scale you should be able to differentiate. If there is a difference, it's more obvious, but in regular scale, it's hard to say. There's a lot of difference. Letter B shows the growth of the wild type versus 836. So 836 looks like it's deficient. Letter C, there's the nonpolar strain versus that of wild type at pH 5.5. So the diamonds show the wild type. The squares is 835, and then (837) are the X's, and the triangles are 836. So 836 is most affected by pH 5.5.
Mm-hmm.
And the final image, growth of the deletion of 836 versus wild type. And you can see 836 is really affected at pH 5.5. Next, then what they do is they show oxidative stress conditions and show that the mutants. This is kind of a strange experiment. Let me just show you from the figure. So in A, what they're doing is they're not adding an oil overlay.
Yeah.
So without an oil overlay, they're saying that that's oxidative. That's stressed by.
Yeah.
Yeah.
Oxygen. Got it.
And letter B, what they're doing is they're adding paraquat to show if there's a difference.
Yeah.
And there seems to be a difference, but it's a slight difference.
Mm-hmm.
Yeah. Next, what they do is they try and look at the biofilm formation, and they say that if you look at the deletion of 835 and 836, that affects the biofilm the most.
Mm-hmm.
If you look at the competence genes, what they're showing is that I don't have it mentioned here, but in the paper they say that the expression was also followed for COM-E.
Mm-hmm.
I think that's correct, COM-E, and they saw no expression change with COM-E.
Mm-hmm.
However, with COM-YA, there is a marked decrease, and with COM-X, a marked increase.
Mm-hmm.
And they feel that the system works to the COM-YA system through COM-X.
Mm-hmm.
Here's more expression data, just trying to find the details of the different expression levels. what they're testing are the various genes here. So RelP is tested in A, and RelP in the deletion mutant for 835 is really downregulated. For 836, it's up to almost wild type levels. But if you, that's for the non-polar mutant. But if you take a look at the polar mutant, it's downregulated.
Mm-hmm. Okay.
Letter B, if you look at the RelRS system, the RelRS system in the deletion mutant for 835 non-polar mutant
Mm-hmm.
is down as well as it is for the 836 polar mutant.
Mm-hmm.
Letter C, if you look at just the genes of 835 and you are looking at a deletion for RelP,
Mm-hmm.
again, the 835 gene is downregulated.
Mm-hmm.
Oops. 836 downregulated in the same 3 gene deletions. Okay, and last figure from the paper, they're also
Hold on,
making...
go back again.
Sure, go ahead. What would you like?
Oh, this is real. These are the mutants.
Mm-hmm. Or these are the genes looked at, and these are the mutants up here.
Oh, I see.
So at the top, we have wild type 8 deletion of 835 non-polar mutant, 836 non-polar, 836 polar, 837 non-polar, 839 non-polar.
835 polar 6. So it looks like 837 has an effect on RelP. And
836 and 835 non-polar have an effect on RelP because they're so down.
non-polar but polar.
Oh yeah, there's a difference between polar and non-polar, right?
So then you need to knock out both.
They also do the 835 non-polar. I'm not sure why they didn't put it here. They do show that the non-polar is different than the polar.
Mm-hmm.
Back in the table, the expression table.
Mm-hmm. Okay.
Okay. So all of this, what they're thinking is that the alarmone, guanosine pentaphosphate, is affected because it's under control of RelP, correct? Then what you'll want to do is find out how much alarmone is made. So here you have the pentaphosphate TLC on a radiograph, like a radioactive TLC. So you can see the spots for just GTP for the triphosphate, 4 phosphates and 5 phosphates. And now if you follow the 5 phosphates in wild type, you get a nice dot. And if you delete the RelP, you should get no spot for the guanosine pentaphosphate because it's deleted, right?
Mm-hmm.
So it should be reduced, and it is. And now if you take a look at the 835 non-polar mutant, it's deleted as well. It's reduced as well as 836, showing that under no oxidative stress, you're getting a reduction in PGPP. And that's that paper.
Why wouldn't they do it also in
Oxidative stress. They didn't say. The explanation of this result is kind of short.
Right?
Could be, yeah.
Because RelP here seems to come back a little bit.
Which is strange because that's the deletion mutant. There shouldn't
Yeah.
be anything. So yeah, maybe because it comes back a little bit, it'll probably come back in these other 2 deletion units as well.
Okay.
Okay, next paper. All right, so it's a review paper on the MAR-R protein.
Yeah.
Now, Pereira talks about the MAR-R protein because it's a transcriptional regulator, and most of the work that they're talking about is in E. coli.
Yeah.
So it's named MAR because it's the multi-antibiotic resistance. It dimerizes and represses the activity of MAR-R. It finds the inverted repeats in front of MAR-R where it dimerizes to. Here's the sequence alignment, and the sequence alignment shows that a number of different bacteria have this even positives. Okay, so there's also the winged helix turn helix, and the wing are the 2 beta sheets, beta 2, beta 3, and the 2 alpha helices, A3 and A4, make up the 2 DNA binding domains. The alpha 3 and alpha 4 in between would be the turn. So alpha 3 orange, turn brown, and then so if you go orange, turn brown, blue, blue. Orange, turn brown, blue, blue. So this forms the DNA binding domain. And they've got the crystal structure for both Mycobacterium as well as Deinococcus radiogerans. The reason they included radiogerans is because it's got an extra region attached to it. Also, oftentimes the protein structures don't resolve in the double beta sheet because there's a lot of unstructured. Yeah.
Got it.
Next, in the MAR-R protein, this picture just shows the region that it interacts with the DNA. So you've got the alpha helix 3, that's the helix, there's the turn, alpha 4, turn, helix, and then the winged domain for beta 2, beta 3. So the gold region right there represents the DNA binding domain in the major groove, and the minor groove, it's the wing.
Cool.
And they can show this. So MAR-R has one homolog, MTH313. This homolog is bound to salicylate, showing the drug binding domain. And it's also done in TCAR, where the salicylate's inside of that drug binding domain. And what they're showing in this image is they're showing that when it's bound or not bound, there's a bit of a conformational change due to the loss of the ligand in the structure. Next, what they're showing is the disulfide bonds are important. And that's the paper, I think. Yep, that's it. Thank you. We did record it, didn't we? No. Don't tell me. Control Shift.